All right, thanks for the introduction. I'm Bernard. I'm, again, a grad student in Kirk Lomulo's lab. You can tell who he is because we're wearing the same shirt today. Totally not planned. And I want to talk about some work we've been doing in our lab to estimate the distribution of fitness effects of new mutations. All right, so the distribution of fitness effects, or DFE as I'll refer to it, refers to the effect that each new mutation has on an individual's fitness. So the reason why we consider this distribution so important is because it quantifies the relative rates of sort of strongly selected versus weakly selected or neutral new mutations entering into a population. So strongly deleterious mutations or strongly selected mutations are quickly removed by natural selection, but weakly deleterious or neutral mutation may persist if genetic drift brings them up to appreciable frequencies. So the quantification of the DFE is actually a major objective for population geneticists and key for understanding genetic diversity. Before going on to talk some more about the DFE, I just want to introduce this summary statistic that we are going to use in our analyses. And it's called the site frequency spectrum, or SFS for short, as I'll refer to it again from now on. And the SFS quantifies the number of sites at a particular frequency in the data. So for example, in this sample data set here, laser pointer kind of sucks, but you can see that there's 1, 2, 3, 4, and 5 sites at frequency 1, quantified here in this histogram, and so on and so forth. So the site frequency spectrum is a useful summary statistic because it can be directly linked to a population's evolutionary history. So in this cartoon example, I'm showing you 3 different neutral SFSs, and in gray is what you would kind of expect if you had a population of constant size. And then we can see that different population demographies, such as an expansion, so shown here in red, or a population contraction, shown here in blue, will skew the SFS in different ways. So we can learn something about demography by looking at these differences. We also know that selection changes allele frequencies. So this figure here shows how deleterious variants are expected to be at lower frequencies than neutral ones and how that affects the SFS of selected sites. So the SFS for selected sites should be shaped by demographies similar to neutral sites, but in addition should be shaped by selection. So using this knowledge, we can establish kind of a framework for quantifying the effects of selection. If we have these sort of 2 predetermined classes of sites, we have a selected class and a neutral class. And since we're using coding sequence for these analyses, we're going to assume that synonymous sites are neutral and that non-synonymous sites make up our selected class. And any difference between the selected class, or the non-synonymous SFS, and the neutral class, or our synonymous SFS, is going to be due to the effects of selection. So this thinking, isn't novel. It's actually been used in a couple of earlier studies to quantify the DFE, but these studies have been particularly limited because they're from, I think, 7 or 8 years ago and quantify the DFE in much smaller genomic samples. So I'm showing you 2 of these estimates in blue and black, and these estimates are very popularly used in the human literature. And I've bin the SFSs sort of by range of selection coefficient, and I'm going from neutral on the left, and as you move to the right, more strongly deleterious stuff. So again, as I said, these studies quantify the DFE from smaller data sets, and we know that deleterious variation should be at rare or low frequency in general, and there should be something to be gained from using larger data sets, which in theory should contain more more deleterious genetic variation. In fact, there's another study. So if you'll notice, this other result is very strange if you look at the 2 previous ones, but this study looked at a larger data set of 200 individuals or so and found a DFE that was completely different from these previous estimates. This estimate's kind of flown under the radar, I think, in the human literature, but we saw this result and we essentially wanted to quantify or to get to the bottom of why this estimate from a larger data set was so different from two previous estimates from smaller data sets. So the first objective of our study was to fit a DFE to some larger data sets. So we downloaded a few different data sets, these more recent, larger sample size, did some data processing, and got these sample sizes, which are quite a bit bigger than these older studies I mentioned. And then we wanted to find a way to fit a DFE to these data sets. So there are a lot of different options that exist, and I've listed some of them here, but they all have some serious limitations when working, especially with big data sets. So they're very limited in what they can do. For example, they can't account for complex demographic scenarios. Some just don't work for large samples. One of them in particular really annoyed me because you have to click through a GUI and then it crashes randomly. So you can imagine how frustrating that gets when you're trying to run it on simulations. Of these, there's this program called DADDY by Gutenkunst and colleagues. And it seemed to be the closest to what we wanted, but it's originally designed for demographic inference, not for inference of the DFE. You can do a lot of sort of convoluted arm twisting and get it to fit a DFE to small samples, but it actually becomes really, really slow and practically unusable when working with large samples. So this brings us to the second aim of our project, is to actually fit a DFE to these larger data sets. We needed to develop something that would get that job done. There are a couple of additional assumptions in this inference framework that I just wanted to clearly state before we move on. I'll go into the details in a little bit, but the assumption is that all sites are treated independently, as in there's no linkage between any SNPs in our data set. Then each entry in that site frequency spectrum I told you about can be treated as an independent Poisson random variable. And this allows us to easily use a maximum likelihood approach to estimate our DFE. Okay, to provide a little more detail, I'm going to just explain quickly this following equation. And we use this equation to calculate an expected SFS given some demographic model and some DFE. First part of this equation deals with binomial sampling, and we just use this part to get from population allele frequencies to sample allele frequencies. The next part in this equation is computed kind of straight from DADDY, and it's the population allele frequencies given some demographic model and a single selection coefficient gamma. And the last part of this equation deals with the DFE, which actually can be any arbitrary distribution that we specify. So with DADDY, we fit a demographic model first to the neutral class of sites, and I won't go through how to do that, but it's almost exactly like this process, just without adding the DFE terms. And then we fix the demographic model to these estimates, and we generate an expected SFS for that fixed demography and a range of single selection coefficients. And then we essentially take weights from the DFE and compute a weighted average or integrate out the DFE. Since we assume that the SFS is composed of independent Poisson random variables, we can then just sort of plug and chug here to compute the likelihood of our data given our demographic model and our DFE. And we then search for the sort of parameters of the DFE that maximizes this likelihood, and that's relatively straightforward, but as I mentioned earlier, DADDY is really slow when dealing with large samples, in large part due to the fact that you have to sort of compute these SFSs during each step of the optimization, and there's no way to sort of make this faster in the standard implementation of DADDY. So to address these problems, we modified kind of a lot of the inner workings of DADDY to be more DFE friendly. And the major usability update was that we, instead of computing the SFS during each step of the optimization, just did everything at once and then just reused it for each step. And since we fixed the demographic model and only changed the parameters of the DFE, it seems to work fine. We also made some other changes, but I don't really want to go into the details except to say that we also added some options to allow people to use constrained optimization if you want to fit very complex mixture DFEs. And finally, one concern that we had with this sort of framework is that things that we don't model explicitly may affect the end product of our inference. Specifically, our concern was that in these larger data sets, yes, you have linkage, and you also may be capturing some aspects of population structure that you wouldn't maybe in a smaller data set. So we did a ton of simulations to try to see if this did have any effect on our inference, and we found that even though our sort of demography, so on the left here, was a little bit off, our DFE was still being recovered properly. So yeah, basically our simplified models to approximate the effects of population structure and link selection seem to work. Okay, so verifying that everything worked, after that we actually fit stuff to those data sets that I mentioned earlier. And in black and gray here, I've depicted sort of the older estimates, again, binned in terms of sort of range of selection coefficient, going from neutral on the left and getting more deleterious as I move to the right. And in the various shades of red here, I'm showing you our estimates from these larger data sets. So the sort of major difference between these older estimates and our estimates are that we predict fewer strongly deleterious variants in this last bin here, and more nearly neutral new mutations as shown in this first bin here. In addition, I haven't shown you that other sort of funky result from the study by Lee and colleagues, but the DFEs we find don't really support their conclusion. So we speculate a bit on why that is in the paper, but I won't go into detail here. We also find that mixture distributions fit better to large data sets. So previous studies used just a simple gamma distribution to sort of approximate the DFE, and our results suggested that a mixture distribution composed of a point mass of neutral mutations and a gamma distribution fit better to the large data sets. Again, we did a bunch of simulations to investigate why, and it seems like for small samples, increasing the complexity of the DFE does not improve the model fit, even though the simpler DFEs are actually wrong. And this is just another example of why sample size seems to matter for this kind of inference. So our results overall, they seem pretty similar, but they may have subtle consequences for human population genetic models. So if you remember earlier, I said that strongly deleterious mutations are sort of quickly selected out of the population, regardless of the effective population size. But if you have a DFE that has more weakly or nearly weakly selected or nearly neutral mutations, then those mutations may persist in the population due to increased genetic drift. So population genetic models which use the earlier DFEs may be sort of subtly under-predicting the amount of expected levels of genetic variation, but especially for younger variants. This is because their results predict more weakly deleterious variation again, which is slower to be removed by selection. There's also somewhat of a controversy about the distribution of deleterious variants amongst different human populations, again, due to the differences in demographic history and effective population sizes amongst different humans. And again, our DFEs suggest that humans overall may have an increased mutational burden, again, because we have more weakly deleterious mutations. I just also wanted to quickly plug some work that my lab has been doing. So we've taken this method and used it to investigate the differences in the DFE between species. And if you're interested in why, check out this paper by a postdoc in my lab, Christian. And of course, if you want to know more about our project, you can check out the paper. It's out in genetics now, and the code's available on our lab GitHub. And with that, I'd like to thank you for your time, and I also want to thank Kirk and Christian. They were awesome to work with in this project, and my lab members for all the important feedback they've given me. And thanks.